Não? Oi, 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 oi. Já? Alto? Oi, oi. Tá. Good afternoon, everyone. My name is Lidiane Lúcia. I am a doctor in forest science and research and seed plant propagation at the National Institute for Amazon Research in Brazil. I am a coordinator in BioSeed project. My friend distributes a card of my project and chocolate from Amazonas for you. And before I talk about my research, I want to do a little presentation about Brazil and Amazonas, because sometimes people, when thinking about the Amazonas and Brazil, think only stereotypes. And Brazil is much more than football, carnival, and beaches. Brazil is a continental country with a diversity and richness. We have 60 major biomes, a wide range of climates, and one of the richest biodiversity on the planet. We have the Atlantic forest, Caatinga, Pampas, Amazon, Cerrado, and Pantanal. And within Brazil, Amazonas is the largest state located in the north region, and it's home to largest portion in the Amazon rainforest. Many people imagining the life in Amazonas happening on the isolated forest community surrounded by untouched jungles. But Amazonas is also a place for university, advanced scientific research, innovation centers, and growing bioeconomy initiatives. And yes, we have the largest rainforest in the world. Amazon rainforest stores enormous amounts of carbon, regulates rainfall patterns across South America, influences global climate stability, and shelters unique ecosystems with millions of species, many of which are still unknown to science. However, this immense natural health faces a serious and urgent threat: deforestation. Deforestation in the Amazon is driven by multiple factors, including the illegal logging, unsustainable land using, mining, and agricultural expansion. And the deforestation goes far to three laws. It leads to biodiversity decline, soil degradation, water imbalancing, greenhouse gas emissions, and disruption of ecological services that support both local populations and global climate regulation. We see the picture the Black River in November 2022, and after one year, the same Black River in November 2023. And the center of restoration, there is something small but extremely powerful: seeds. Native forest seeds are the foundation of ecological restoration. Without high-quality seeds, we don't have successful and regeneration, no recovery, degraded ecosystem, and no sustainable seedling plants. However, is the lack of information about native seeds. Many species still don't have establishment protocols for viability analysis, storage, and germination. Today, these protocols are created by Ministry of Agriculture in Brazil. And this is especially critical because most Amazonian forest seeds are recalcitrant. This means they cannot be stored for long periods and lose viability rapidly, which means the fast, reliable, and non-destructive testing methods are urgently needed. In this example, we see the seeds of Onelcarpus bacaba take a leave 5 months to seed growing to seed growing. And today, this is the point of my presentation today: viability of native forest seeds to grow the images, contributed to forest restoration in the Amazon. All research address on the main limitations prevent the wide use of native species in the restoration projects, the difficulty of evaluation seed quality efficiently and accurately, because the traditional seed testing are slowly, expensive, and they don't have seed testing protocols to species from Amazon. And we collected 20 native forest species in 3 seed collecting areas distributed in different ecosystems from Amazons. We have 11 seed collecting areas monitored by Bioseed project. And we use 2 main tools: X-ray images and ground eye scanner imaging. X-ray to observe internal structures such as embryo, integrity, and reserve issues, and ground eye scanner imaging to access external morphological traits such as size, shape, and texture. And we correlation all data to do algorithms to predict the seed viability by images, the integrity new technology in research with native forest. And we integrate all the data, color, internal imaging, dimensions, texture, and germination percentage. Oh my God. Sound is right, you know. All results shows that cotyledony is very important for exobuminose seeds. Like this picture shows RGB and X-ray images of Hermosia seeds, and A, we see the viable seeds, shows the complete internal filling, and B, no viable seeds, shows the presence in internal crack, and C shows the crushed seeds with uneven internal padding. Look it down again. And here we see RGB and X-ray images of Cariniana legali seeds. And the image A, we see cotyledon presence, and B show the cotyledon absent. And they show, and A and B show the difference and the RGB image. And there we see the X-ray image of Diocla violacea seeds, dentail, and the positional elon, and the seed coat. And shows the viable seeds, and B and C shows the seeds with larva completely, and C completely deteriorated seeds with larva. And here we see X-ray images of Salmonella tubulosa seeds, which the tail show the embryo presence. And another result shows the importance of Haustorium structure in monocotyledonous seeds. Here we have Oneocarpus batala X-ray images before soaking in water. These species have a lignification endosperm, can be a false damage, but it's not damage, it's the characteristic of a lignificated endosperm. And this picture shows after soaking X-ray images, and A is a no viable seeds and B viable seeds, and show the ways, pathways, and here the germination bottom, viable seeds. And the most important result shows the correlation, more than 90% correlation, between seed viability by X-ray imaging and seed testing. This means imagining analysis can really predict seed viability without destroying the material. Another result shows the algorithm construction in RGB images, which this machine, ground eye scanner. Here we have a decision network of viability Heterolobes-Coburg, using biometrical parameters VNA, and these classificate seeds by the diaspora type. And we take more 300 morphological data points, and PCA do adjustable variables for each species by the diaspora type. We do training the algorithm with X-ray images in the R Studio program. We made 13 radiographic analysis protocols today, before the traditional seed testing can take even months to predict the viability, to see the viability. 5 months, 6 months. Today, with this project, with the help of artificial intelligence, in 40 seconds, we know if the seed is viable or not viable. This is an important step toward the use of artificial intelligence and automated image analysis for native seed quality control. It reduces the low genetic resource, improves restoration success, and strengthens sustainable forest management strategies. Ultimately, protecting the Amazon is not only about preserving trees, it's about preserving process, relationships, and the futurist possibilities. And sometimes this future begins with something as small as a single seed. By improving how we understand and evaluate native seeds, we create stronger foundations for restoration, biodiversity, conservation, and climate resilience. This is my research team. I cannot do it alone. This is my students' post-graduation, my friends' research, and I do this with together 2 universities, Federal University of Amazonas and Federal University of Eastern Pará. Thank you very much. This all Instagram, please follow us. And I look forward for any question. Thank you very much for the opportunity.
And I would like, okay, and now I would like to
Hello, thank you for having me today. I'm Hanna and I work at the Institute of Dendrology, Polish Academy of Sciences. And today I would like to share with you our work on seed longevity at molecular level based on model system for comparative seed biology. But before I will get to the topic, a brief explanation why we are even interested in the seed biology. As you can see here, the Judean date palm seeds are the oldest known seeds that have germinated and are growing to this day. They have been found at the palace of Herod the Great at Masada, on Masada in Israel, and they have been kept in cool, dry place and germinated in 2005, and you can still see the date is growing. But this is the exception. Seeds over time lose the viability and germination capacity. So this is the topic that interests us in the Institute. Seeds are pivotal for plant reproduction and dispersal and also for biodiversity. So to safeguard the genetic resources, so-called gene banks have been established where seeds are kept under strictly defined conditions. These are the seeds of specific genotypes or endangered species, or they are a basis of our diet. And the best known seed bank today is the Svalbard Global Seed Vault, which you can see here. Probably you know this picture. However, the biggest is the Millennium Seed Bank Partnership coordinated by the Royal Botanic Gardens in the UK. They are holding over 40,000 plant species living on Earth. But The seed loses viability is one of the greatest challenges for gene banks. This is why at the Institute of Dendrology we investigate the genetic basis of seed longevity. As you probably already know after Mikołaj's lecture, seeds are categorized into 3 distinct categories: the orthodox, intermediate, and recalcitrant. And in the Institute, we are interested mostly on seeds produced by Acer platanoides, Fagus sylvatica, and Acer pseudoplatanus. But today I will talk about those 2, the Norway maple and sycamore maple, which are 2 closely related species, but they produce distinct categories, seed categories. Norway maple represents orthodox and sycamore recalcitrant. Their natural range overlap to a large extent. As I mentioned before, they are 2 closely related species and 2 distinct categories, which makes them the perfect model system. And also, their physiology is already well known and established in the literature. In our institute, we have a rich history of researching the physiology, biochemistry, and dormancy, maturation, and aging of seeds. What makes seeds desiccation tolerant? There are several factors. First is storage reserves, presence of chaperones, seed coat, metabolic intensity, active antioxidant system, and all of these factors are regulated by the abscisic acid-dependent gene expression. We are mostly interested in the active antioxidant system, and in particular, in the protein thioredoxin, which participate in at least 12 mitochondrial processes, ranging from energy production, metabolism, to protein synthesis, stress responses, and interorganellar communication. Thioredoxins also act in mitochondria as molecular chaperones under oxidative stress. These proteins activate other enzymes by the active site, which has 2 cysteines. And those 2 cysteines create disulfide bonds. And thanks to those disulfide bonds, they can create a complex with a target protein. So these proteins are well described in the animal kingdom, but still there is less attention in the plant kingdom, especially in the seeds redox system. So we asked ourselves, does the profile of thioredoxin target proteins differ between seeds belonging to different categories? And we checked this by affinity chromatography, and in the result, we identified 238 proteins in sycamore maple and 69 in Norway maple. But these numbers reflect detectability, not the network site. The sycamore maple has active metabolism and rich proteome. In contrast, Norway maple has quiescent metabolism and sparse proteome. Targets are likely present but below the detection. Next, we categorize the proteins based on Keck classification. And we found out that we have proteins that are involved in environmental information processing, metabolism, cellular processes, genetic information processing, and some of them are unclassified. So both proteomes were different. We saw that the 41% of the sycamore maple proteome were proteins involved in metabolism, 38 were genetic information processing, 9 were environmental information processing, 5 were unclassified, 7 were cellular processes. In contrast, in Norway maple, almost half of the proteins were associated with genetic information processing, 24 in metabolism, 22 were unclassified, and 7 were involved in cellular processes. The main picture in sycamore maple are the proteins involved in metabolism, particularly in ATP synthesis and the Krebs cycle, but also in stress response, like chaperones, including CLIA proteins. In in Norway maple, we see the metabolic quiescence, and and investment in transcription and translation control. We complemented these studies with the gene expression analysis of 3 genes that are involved in the thioredoxin redox system. The first is obviously the thioredoxin, and we see 2 distinct expression patterns. You can see in sycamore at the beginning of the maturation and at the end of the maturation process we have 2 peaks, and in contrast in Norway maple we have 1 peak about 1/3 of the maturation process. Next is 1,cis peroxyredoxin, which also has distinct patterns with a higher expression in sycamore maple. And you can see that in both species, the expression is relatively stable during the maturation process. And last but not least is the thioredoxin R anti-RA reductase, which also have distinct patterns, and 1 big peak in the 19th week after flowering in sycamore maple. We also checked how the expression patterns differ during the storage. And as you can see, only sycamore maple have a detectable expression profiles. And in Norway maple, the expression of 1,cis peroxyredoxin and the reductase are very low. Here we can see anything. And in sycamore, the expression declines during the storage. So to see if this decline is related to viability, we made the oxygen consumption rate measurement, which is a proxy of viability. And we checked it on fresh 2- and 5-year stored seeds. On the left, you can see the orthodox seeds of Norway maple, which retain respiratory competence at 2 years, and despite near silence transcript of the thioredoxin system. They slightly lose the viability, but probably if we would make a germination test, they would still be viable, because you can see that here they are quite stable. And in contrast, the sycamore maples, after 2 years, they already losing viability. The OCR drops quite dramatically here. And after 5 years, we see that they are starting from the high point, and they are dramatically lower through the measurement. So summarizing, the higher number of thioredoxin targets identified in recalcitrant seeds compared with orthodox most likely reflects the metabolically active state and the richer proteome of recalcitrant seeds rather than a more extensive redox network. Orthodox seeds contain homologous targets at levels below detection at this developmental stage. Within each interactome, however, the relative cake composition is consistent with seed strategy. In recalcitrant seeds, metabolism-related targets, and the orthodox target set is comparatively enriched in genetic information processing in line with metabolic quiescence and a quality over quantity protein strategy. Thioredoxin transcript level remains low during maturation in both species, but with species-specific peak timing. And 1,cis peroxyredoxin transcript is detected throughout maturation in both species at stable levels within each species, but consistently higher in the sycamore maple. Among the 3 transcripts, the reductase shows the largest species-specific differences in both magnitude and dynamic across maturation, pointing to the reductase as a key category-specific layer of regulation in the thioredoxin system. After 2, 3, and 5 years of storage, all 3 transcripts decline toward near silencing in both species in recalcitrant seeds to progressive loss inconsistent with viability decay, also reflected in oxygen consumption rate. And none of this would be possible without all of those people, so thank you very much for cooperation, and thank you for your attention.
Dzięki bardzo.
Jest to, nie?
Dokładnie. I co, jak to, co? No, jakby tak, no. Well, hello, my name is Katarzyna Kondrat. I am a PhD candidate at Adam Mickiewicz University, and today I will tell you about the seed number, seed size trade-off in European beech. So as we all well aware, our life histories are shaped by trade-offs, and that both applies to plants and animals, including as humans. Like, for example, if last night I've spent more time on this presentation, it might have looked better, but at the same time, that would come at the cost of my rest. And I really like my sleep. And when it comes to all trees, they also face some choices that come from the fact that the resources are finite, which means they can't really equally distribute them between different life functions such as growth, storage, and reproduction. And let's say in one scenario, more of the resources go to growth, which makes the trees maybe more competitive, but at the same time, that comes at the cost of reproduction that most likely is hindered during that time. Then in different scenario, maybe more resources go to actually growth, to reproduction, which means that more offspring is produced, but that comes at the cost of growth and storage. And today I would like to focus on reproduction and on something that I like to think as a sort of a trade-off within a trade-off. So that is a classic reproductive trade-off that was first described in this paper by Smith and Fretwell from 1974, where they pretty much came up with a model that was finding the optimal balance between the size and the number of offspring. And that model can be both applied to animals and plants, but so it happened that plant ecologists really liked it, and it became sort of a getaway to what is now a very broad literature on the seed size and seed number trade-off. And at its core, the idea is quite simple. Since the reproductive budget is fixed, the resources are finite, plants pretty much only have two scenarios. They can either invest less per seed, which means they'll produce many small seeds that maybe will be at greater odds of escaping predation or getting dispersed, but at the same time, they aren't as well provisioned. And in different scenario, the investment per single seed is bigger, which means that seeds are better provisioned. They most likely will germinate better, but their number is smaller. And that is all very neat and clean and simple, but it actually gets more complicated when we talk about mass seeding. And when we talk about mass seeding, we talk about this phenomenon in which plants in a given population produce seeds with a very large intra-annual variation. And that can apply to herbs, but also shrubs and trees, just like European beech. And for example, here you can see that in some years there are virtually no seeds being produced, whereas the next year the seed production can reach over to 400 seeds per individual, for example. And very often during mass years, and we know that when it comes to European beech, that the resources get allocated to reproduction and the growth during the mass years is hampered. And having all of that in mind, we sort of like face a puzzle as of what actually shaped the per seed investment in this species. Because, well, on one hand, we have a trade-off, but we also have masting, which complicates things, especially because when we think about masting as this period when all the resources go to reproduction, and masting is supposed to maximize the reproduction, the production of viable seeds, the seedling emergence, it wouldn't really make sense for a tree to produce lots of seeds that, well, aren't well provisioned and are mediocre at best. So we thought that maybe when it comes to masting species, the classic trade-off wouldn't really apply. And the third element are the macronutrients. When it comes to temperate trees, and especially European beech, we know that this species is, its seed production is nitrogen limited, which means that actually the per seed investment when it comes to protein can be smaller, whereas carbon limitation doesn't really play that much of a role. And last but not least, we also have climate. And even according to the classic framework, per seed investment can vary from different climatic conditions, and most likely is better when the conditions are more favorable. And having all of that in mind, we came up with a set of predictions. And well, first of all, when it comes to seed mass, we figured there are like two different scenarios. First of all, the seed mass can be maintained if the reproductive allocation actually increases during a mass year, or if we stick to the classic trade-off, it can actually decline. On the other hand, when it comes to seed composition, since European beech is nitrogen limited, protein content may decline with rising seed production. When lipid content, we thought it should vary less. And last but not least, we also figured that the seed provisioning would most likely be better in more favorable climates with longer growing seasons, for example. And we would see lower seed mass and nutrient content at species branch margins. So in order to do that, we collected seeds from 123 sites across the whole species range in Europe during 2 years. And in the main analysis, we ended up with 93 sites with almost 2,000 trees because they were the only sites where the sound seeds were found and which size was then measured. And for the seed chemistry bit, we ended up with 33 sites, almost 500 trees. And the sampling method is actually, well, quite simple. So it uses a standardized 3.5-minute search under each tree canopy. And later in the lab, all those seeds are being sorted, and we discard the predated seeds and the unpollinated seeds, and in the end, only the viable seeds were weighed. And then next, those seeds were also analyzed when it comes to macronutrients. And for the analysis themselves, as the tree-level predictors, we chose the seed production, so the total number of seeds per tree, tree size, which is DBH, diameter at breast height, and the shading class. For the site-level climate, which was growing season length, and within that growing season, we calculated mean temperature and also mean precipitation. And as a response, we fit 3 separate models for seed mass, protein content, and lipid content. And as it actually turned out, we saw no seed size number trade-off. So this graph shows the relationship between the number of seeds produced per tree and the seed mass. Each point is an individual tree with its confidence intervals. And as you can see, the seed mass actually increased, and it increased by 14% between low and high seed production. When it comes to macronutrient content changes, panel A shows the relationship between seed production and the percentage of protein in seed dry mass. And again, each point is one tree. And as you can see, when it comes to protein, actually the protein content declined by over 30%, which stands in line with the fact that European beech is actually nitrogen limited when it comes to its seed production. On the other hand, lipid content increased with seed production by 14%. And last but not least, we also looked at how seed mass changes across species range. So map A shows the changes in seed mass, map B in protein content along the species natural range. And as you can see, the both seed mass and protein content actually increased in the Atlantic and Atlantic temperate regions of Western Europe. So those are the places where precipitation is quite high, growing season is long, and the temperature are mild. And both of those factors actually decreased towards the southern margins of the species. So the places where growing season is long, but the temperatures are high. And at the same time, both of those were also quite low in the mountainous areas like Alps and Carpathians, so the places where both temperature and growing season are low. So to sum it all up, the seed mass was actually maintained at higher reproductive output, which kind of contradicts the classic framework. But what did decline was the protein content, which stands in line with the nitrogen limitation in European beech. But what didn't decline was the lipid content. And when it comes to climate, it also did have an influence. And it points us to the fact that those climate-related reductions in seed provisioning can actually constrain regeneration at the range margins, especially at the southern range of the species. So thank you for your attention. I especially like to thank my whole lab and all the collaborators across Europe who made it possible by collecting the samples. Thank you.
Thank you very much. And the next presentation will be presented by Amir Mokhtari. Please.
Good afternoon, everyone. My name is Amir Mokhtari. I'm a PhD student here in Kórnik in the Department of Developmental Biology, Institute of Dendrology, Polish Academy of Science. Today I want to talk about our recent study published in Nature Scientific Report entitled with Photosynthetic Pigments in Developing Seeds of Acer platanoids and Acer pseudoplatanoids. The colleagues already talked enough about these two species, the differences, but I will also go deeper and tell you a story of chloroplast behind seed longevity. So let's just start with the central question. Why do two green maple embryos behave so differently after maturation? If you open the seeds of these two species, you will see, and removing testa, you will see that both species are green. We call this kind of embryos like as a chloroembryos. It doesn't mean that when they are dry, they can do photosynthesis. Just being green color, we call them chloroembryos. Acer platanoids, known as Norway maple, so categorized as orthodox seeds, so it's desiccation tolerant. Because of that, it's longer lived. On the other hand, we have Acer pseudoplatanos, known as sycamore. So it's recalcitrant seeds. It's used viability after like 2-3 years, but and also categorized as desiccation sensitive. So it's shorter lived. It's like the previous Katarzyna mentioned that it's a trade-off. So just we want to compare them, like because of being this desiccation tolerant, so they have some differences between their amount of chlorophyll which they have. Acer platanoids has stronger pigment decline. I will go deeper in next slides, and signs of structural chloroplast changes. But the next one, sycamore, has the opposite side. Less pigment downregulation, and photosystem 2 remains potentially active at key stages. So we hypothesize that maybe seed longevity is somehow related to the amount of chlorophyll, not only at the final stage of seed maturation or drying stage, also analyzing the chlorophyll amounts during seed development process, like seed morphogenesis, embryo morphogenesis, and maturation, could tell us some information about seed longevity, how they will behave after maturation. Just I want to, of course, all of you are experts, just I want to elaborate in chloroplast structure. So this is chloroplast, and if we go deeper, we see the thylakoids and granum structure. In this area of the thylakoids, we have thylakoid membranes. So this is the place that all photosynthesis occurs. So, and the first responsibility is on photosystem 2, which this chlorophyll, mainly chlorophyll A, B, and carotenoids are located. So this photosystem 2, this green protein that you can see there, it's a protein antenna, which the place that all pigments are located. Here we have chlorophyll A and B, But and also the orange color that you can see there, it's the carotenoids. So chlorophyll A and B, they just you see that they are look like each other. Just the difference between them is that chlorophyll A just change one decarboxyl group to one aldehyde groups, and they become chlorophyll B. And this is very interesting for me. Sorry. It's very interesting for myself because how the seeds, how the plants like evolve themselves or understood by themselves that we have chlorophyll A, it's not enough for us. Let's create another chlorophyll type. Why? Because chlorophyll A would absorb specific wavelengths of the light, but on the other hand, they were losing some other wavelengths. So they adapt themselves to absorb other wavelengths. So by creating chlorophyll B and also carotenoids. So why pigments matter in seeds? It's unknown the reason that the seeds can receive all chemicals, all nutrients from mother plants. So why they want to still do photosynthesis? It's unknown. Maybe they want to like locally produce their needs, like their proteins, their like sugar, everything by themselves. So this is the first responsibility of these pigments. Chlorophyll A and B capture light energy and are bound to chloroplast thylakoid membranes. So by doing photosynthesis, developing embryos can perform photosynthesis producing oxygen, ATP, and NADPH that support seed metabolism. In orthodox seeds, photosynthetic structures are expected to be dismantled during maturation and drying. We observed it, I will show you later. But retained chlorophyll and active photosystems in sycamore can increase oxidative risk and reduce seed storage performance. So take-home idea would be here that in seeds, chlorophyll is not only a pigment, it can act as a marker of developmental shutdown, chloroplast remodeling, and potential longevity. I'm not going to get into it, but as a summary, like experimental design, we had 2 species. Sampling was done here in Kórnik in 3 consecutive years. And organs. We separately analyzed embryonic axis and cotyledons of these seeds during 4 stages, starting from embryogenesis, which we had just only possibility to study whole embryos. Then we went to morphogenesis, embryonic axis and cotyledon separated, maturation, and drying stages. As you can see, these 2 pictures, it's the flowers of these 2 species. Like 2 weeks ago, I captured them that you can see that they are green. And even the inside, it's green, they can do photosynthesis. In this study, like we did like normal extractions for analyzing chlorophyll A and B. We measured their pigments, the ratio between pigments, and also we analyzed photosystem 2 activity by chlorophyll fluorescence. And at the end, for visualization, we used light microscopy for the seed structure and confocal microscopy for chlorophyll autofluorescence. Like the thing that we measured, all of them together asked to the main question that what happened during seed development. This is the first figure. It's related to Norway maple, Acer platanoids. And as you can see, from the 12 weeks after flowering, the number that you see there is 12, the weeks after flowering. From the 12 weeks after flowering, just we have a peak in both organs, in embryonic axis and cotyledon. So, but we have very strong shutdown in chlorophyll and chloroplast maybe till this stage. Maturation is done, and this is start from 12 to 22 weeks after flowering. Also, you can see in chlorophyll B, but less drastic increase and also a slow decline in the amount of chlorophyll B. But in contrast, we have sycamore here. Also, it's higher. But if you compare the amount of chlorophyll A, it's like here it's 6.5. Going back to the previous slide, it's 10.5. So Norway maple, they have more chlorophyll compared to sycamore. But also they are the peak we can observe we have two peaks here. One is 8 weeks after flowering and another one between 14 and 15. But you can see also the declining here, we can observe it, but it's not as strong as Norway maple. It can give us a lot of information because at the last stages, which is drying here, but you know that recalcitrancy, we can dry them more than below than 30%. So we should stop it at 30%. Both are the same. Both have the same amount of chlorophyll at the final stages. But these figures tells us that the amount of chlorophyll at the final stage of seeds, it's not only important. Also, the amount of chlorophyll during the seed development is can be important for us. And total chlorophyll A and B together. Here, the differences is much more clearer because you see here that the green dots belongs to the Acer platanoids, Norway maple, that you can see chlorophyll A and B. They have much higher total chlorophyll content compared to sycamore. But at the final stage, as I said before, it's the same. So by observing these figures, we can say that, okay, maybe the amount of chlorophyll, because both of them have the same amount of chlorophyll, so it doesn't mean that they have, the chlorophyll content can affect seed longevity. But we observe during the seed development procedure that Norway maple has very higher peak than sycamore. We can interpret it in different ways, that maybe they just try to increase the amount of chlorophyll in seeds to produce as much as sugar, ATP, and other source for producing protein. Like we know that heat shock protein are necessary for orthodox seeds to tolerate desiccation, okay? They use this energy to produce this protein and other organic matter to be ready for the desiccation time. But on the other hand, sycamore seeds, they think like that, okay, chill, we have enough time for, after storage, we can do photosynthesis. So it's not necessary to increase the amount of chlorophyll that much. Photosystem 2 activity tells us about not quantity of pigments, tell us about the potential function. So again, at the final stages, both species have the same amount of, in this treat same as in the context of photosystem 2 activity, but again, Acer platanoids, a very sharp increase in the photosystem 2 activity, which the chlorophyll A and B and the main electron excitation in photosystem occurs. But very important for us is carotenoids. Carotenoids composition can tell us a lot of information because they are not only a pigment for absorbing light energy, they are also responsible as an antioxidant because also the best wavelengths of the light that the plant can absorb is between 420 to 450 nanometers, but less than it's going to the blue-violet region. It's very oxidative, can cause oxidative damage to plants. But carotenoid can absorb 400 nanometers. So they are responsible also for the photoprotection aspects. Amount of carotenoids in these two species in different stages, like if we compare the carotenoids in two species, we can see that Acer pseudoplatanoids at early stage, in contrast to other pigments, has higher amount of carotenoids comparing to the Norway maple. It's also gives us a lot of things because maybe Acer pseudoplatanoids is faced with a lot of oxidative damages and facing with a lot of reactive oxygen species. At the final stage, the interesting thing is that Norway maple has a sharp increase again compared to sycamore, because I think that it's because of the losing water content, because they have 10% water content. They are going to be ready for losing water for the storage. So that's why we think that carotenoids increase because of that water loss, that they are like trying to prevent more stress. The most important visualization here for us is, as a confirmation of our study, is this microscopy and confocal microscopy images that you can see here, Acer platanoids and Acer pseudoplatanoids in embryonic axis. There is no difference between autofluorescence of them because both of them, they have irregular and diffuse autofluorescence, as well as the cotyledons of Acer pseudoplatanoids. You see that diffuse, irregulars. But here, coming to cotyledons of Acer platanoids, we see they are compact. So being compact gives us very good information that maybe chloroplast, chlorophyll A, B, it's available, but maybe chloroplasts, they are structurally changed. Maybe chloroplasts changed to other kinds of plastids. So this is very good information us, and this is the basis for our current study that we need transmission electron microscopy images to see just the same as like during the seed development, to see how chloroplasts change, even the structure of the granomes and thylakoids. If they change also, or something like autophagy is happening in Acer platanoids, we say that, okay, we are sure that chloroplast has a negative effect for seed longevity. Just to wrap up, that Acer platanoids, Norway maple, strong chlorophyll degradation, large PCI downregulation, compact chlorophyll signal in cotyledons, possible chloroplast differentiation, we are not sure. We need TEM analysis, better compatibility with seed longevity. But in contrast, totally weaker chlorophyll reduction, photosystem 2 remains more active at key stages, diffuse autofluorescence pattern, possible persistence of chlorophyllous cells, reduced compatibility with seed longevity. Why this matter for forest ecology? A lot of people talked about it before. These two species are native to Europe, at least native to Poland. I don't have that much information about whole Europe, but these are two only native species of maple in Poland. So, and their only way to, like the main strategy to reproduce these seeds, these three species, is by seed. So also for storage strategy, for the seed bank or other things, we need to understand how we should behave with these seeds. Maybe we can modify, especially about the sycamore, how we can change the strategy to at least store them much longer than we store right now, because they lose their viability after 3-4 years. Can we do something to make it like for 5, 6, 7 years? And other things like that by gene editing, that it's in my future perspectives. And of course that we will do TEM validation, transmission electron microscopy. But another thing is that can we do something, but it's like look like sci-fi, but can we do something to modify chloroplast degradation pathways to decrease the amount of chlorophyll and chloroplast in seeds just to keep them be able to tolerate the desiccation stresses. But what else we can do? Redox connection. Like we have photosystem to activity data right now, but if we compare with NADPH and ROS production, antioxidant activity, we can have more validation. And testing whether modifying chloroplast, as I said, and pigment degradation or amino acid, amount of protein, which other people have done it. So we can connect all these to come up with a solid idea. Thank you very much, everyone. I also would like to thank my first supervisor, Professor Eva Kalemba, and my auxiliary supervisor, Dr. Natalia Wojciechowska from Adam Mickiewicz University, and my co-author, Andrzej Kowalski. And please, if you have any idea, first before question, please help me to what would be the perspective for this study, and also you can scan this code. This is recently published last month, or paper. You can study it, and of course that we need your feedbacks. Thank you so much.
Thank you.
Hey everyone, my name is William Souza. I'm a PhD student by Federal University of Jequitinhonha and Mucuri Valleys in Brazil. It's a pleasure to be here. It has been a great conference. When my friend told me about this conference, I just thought that this is a perfect opportunity to share these findings, because despite it's a quite simple work, it's a good opportunity to talk a little bit about Brazil, and of course show that even some basic work in the correct context can be really important. And the work that I'm going to present is entitled Combined Effect of Seed Size and Biofertilizer on Initial Establishment of Mulungu, Erythrina velutina seedlings. And well, before moving into the experiment, let me briefly set the context. Brazil is one of the most biodiverse countries in the world. It holds over 56,000 plant species, representing over about 90% of global flora. And sometimes here, talk with my Polish colleagues, my Polish friends, I figure out that sometimes they don't really understand how big Brazil really is. So just for instance comparison purposes, this state right here is the state that I came from, Minas Gerais, the green one. And it's not the biggest state of Brazil. It's not the second or even the third biggest state, but it's still almost 2 times bigger than Poland. So yeah, that's quite big. It's a continental country. And this vast biodiversity also comes with a challenge. Many of the native species still a lack of basic biological information, such, for example, seed traits, such seeds, morphological and characteristics. And this is important that we have seen in so many lectures and work that seed plays a key role on the plant germination and early establishment. So there is a clear need for more target research regarding seed trait studies to address these gaps. And this is exactly the context that where our student species fits the mulungu, that beautiful tree. Erythrina velutina wild, as popularly called as mulungu, it's a um, tropical region native species, widely distributed on Brazil, especially related to seasonal environment, that it's really well adapted to these conditions, so it's also widely used in ecological restoration and reforestation programs, and also has some ethnobotanical importance due to the traditional medicinal use by the Brazilian people, due to the medicinal properties. So the people used to make, for example, tea of mulungu to relax due to the some compounds that is present in the plant tissue, or for example, some flavonoids and alkaloids that can have anxiolytic effect. And so we can see that beyond the ecological importance also have some practical and social importance. But seed rates and early establishment remain poorly understood on these species. And that's where our justification are. So we all know that seed functional traits strongly influence the germination and early establishment process. And also the seed variation may affect the seedling vigor and initial development. And an important tool that is usually used by farmers and also in recent work is the role of the biofertilizer that can modulate these effects but remain insufficient understood. So these relationships are still underexplored on mulungu, and in that way our objective is to evaluate the combined effect of seed size and biofertilizer application on the establishment of mulungu, Erythrina velutina. And well, starting our methodology, the experimental site, the experiment was conducted in a greenhouse belonging to the agricultural sector of Federal Institute of Education, Science and Technology of Bahia, Campus Guanabi. And about the size measurement, the seed size was measured in 3 dimensions: length, width, and thickness, and all the dimensions were measured using a digital caliper. And based on this measurement, 3 block spots were generated that you can check on the figure 2, 3, and 4, each representing the variation of a specific dimension for large and small seeds. And we can see a clear separation between the two groups, with the larger seeds have a higher results when compared with the small seeds. Also, a good uniformity. And about the experimental design, the seeds were subjected for a 60-minute imbibition in a 50% diluted biofertilizer, and the biofertilizer is composed by banana pseudostin, brown sugar, bovine manure, and plant ash. So the seeds were sown in autoclaved sand in 10 liters containers, and the design of the experiment was completely randomized with 4 treatments and 4 replicates. So 15 seeds each, in total 240 seeds. And the treatment were the small seeds with control or without the biofertilizer as SC, small seeds biofertilizer with the biofertilizer, and larger seeds in the same way, the control and with the biofertilizer, LSC and LSB. The data we analyzed using the ANOVA, and then the data was analyzed with the Tukey test with 5% of significance. The seedling evaluation, the biofertilizer was applied weekly via irrigation, again 50% concentration, and the seeding emergence was monitored daily. The emergence was defined as visible plumar hook above the substrate, and the evaluations conducted for 50 days until the stabilization. So the final measurement was the seeding height and dry mass ingredients, and the dry mass protocol with the washing seedlings, and then dry the seedlings in a hot air oven in 60 degrees by 24 hours, and weight in a precision scale. So regarding the results, about the emergence performance, the larger seeds show higher emergence and faster establishment regardless of the biofertilizer. So the emergence with and without the biofertilizer is still higher than the small seeds. And the rule of the biofertilizer was undefined because there is no significant results between the treatment control and biofertilizer. And the same pattern is noticed in the mean emergence days, in mean emergence time, where the larger seeds had less time, so it's a better result than compared with the small seeds. Also in the emergence speed index, that the larger seeds had a higher index. And about the seeding quality, was measured the seedling height, dry mass, and abnormal seedlings in numbers. And the results indicate that larger seeds lead to stronger and healthier seedlings, while the biofertilizer, again, did not significantly alter this pattern under the tested conditions. So in the graphs above, we also can check that larger seeds, again, had better results, higher values in seeding height in centimeters than when you compare with the small seeds. And in dry mass, it's even more evident that the larger seeds had a higher results in dry mass on the early establishment of bulungu when you compare with the small seeds. And in abnormal series in numbers, the small seeds had a higher values that it's of course a negative results when you compare with the larger seeds. So the final remarks, the larger seeds consistently produce more vigorous seedlings than small seeds, with a higher emergence, faster emergence, and greater height, higher dry mass, and a fewer abnormal seedlings. And the biofertilizer application at 50% did not significantly improve seedling performance under the tested conditions. And the seed size was the main determinant of early establishment in the Erythrina velutina. So therefore, selecting larger seeds is recommended for seedling production, while further studies should be optimize the biofertilizer concentration and application method for this species. So that's all for today. Thank you, everyone. Again, it's a pleasure to be here. If to check the reference, just scan the QR code, and here is my contact. And that's all. Thank you.
Thank you very much. And it was our last presentation today. So now we have time for questions. So if somebody would like to ask your colleagues, it's a good opportunity now or later, because I know you are waiting for the maybe excursion. But maybe you have any questions or comments to the presentations. Yes, please.
I have a question for you, Hannah. Do you maybe know if the green seeds with chlorophyll is more common in orthodox or recalcitrant seeds overall? Maybe there's data about this or not?
To be honest, I don't have the exact data about that. Which one has which one has more color embryos? But I know that being a recalcitrant, it doesn't mean that it's a chloroembryo. But if it's chloroembryo and it's chlorophyllos, active chloroplast, or potentially active chloroplast, they are probably, for sure, it's recalcitrants. I know from my Master of Science that, like onion seeds, they are not chloroembryos, but they are recalcitrants. So, yeah, I would, thank you.
Any other questions? Okay, so maybe I assume maybe you are tired, and it's high time to relax, to eat something, and so I would like to thank you for your presentations. Please remember you have an opportunity if to scientifically interact with your colleagues. It's good. I wish you a good time here, for looking at the landscape casters and also the piece of Polish society, but also for scientific interactions. So thank you very much.